All right, so this is an explanation of the DNA scissors activity from class. So if you're absent or if it didn't make sense. So if you read all this stuff, it talks about restriction enzymes. And restriction enzymes are found in bacteria, and they cut DNA strands in predictable places. So what that means is we can use certain DNA or bacterial restriction enzymes to cut DNA the same. So if we have one kind of DNA and another kind of DNA and we want to put them together, we can cut them and then they will stick together. And so if you look at this next page, there are 6 different restriction enzymes that are on this packet. There's more in real life, but these are the ones we're going to talk about. And then I have a sample, 4 little sample DNA strands over on the sheet that I want you to write on. So what I want you to do is I want you to go through and say which one could cut each one of these. So for example, EcoR1 has a certain sequence. It cuts where the arrows are through. BAMH1 is a different sequence. SMA1, HIND3, ALU1, and HBA1. These are named after bacteria. This is E. coli. And so then they all have their own special one, but E. coli is probably the one that you have heard of, and this is one that we get from that. So for this one, it's from the 5 to the 3 prime, it is GAATTC. And so we'll go through and we'll look at these and we'll say, do we find that sequence? So GAATTC, here it is. And so we have this little chunk that matches the EcoR1. And so it cuts it where this arrow is right here. So between the G and the A and the G and the A on the other strand. So what I want you to do on your paper is I want you to cut, not cut literally, but make a little line between the G and the A and the G and the A. And so we'll make this like zigzaggy little cut. And so then this would be a piece of DNA and this would be a piece of DNA, and we could separate those. And so I want you to just, for the rest of those, I want you to just go through these and figure out which one that each one of these might cut. There are 6 here, there are 4 strands. 2 of these are used twice, or one of these is used twice, and one of these strands has 2 in it. So make sure that you find those. So if you flip to the back of the packet, it's on the 5th page. This says exercise 1, and this talks you through doing this activity. So if you go through and it says you'll now simulate the activity of EcoR1, blah, 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 blah. And then, so some of them are instructions, some of them are questions. So this number 2 says, now separate the hydrogen bonds between the cut sites, cutting through the vertical line, separate the 2. And then it says, look at the new DNA ends produced by EcoR1. Are they sticky or blunt? And so that's a question that you would need to answer on the back of this sheet. And so how you figure out what the answers are is you have to look them up. And so for that one in particular, it's right here on the 2nd page. And it says, this type of end is called a sticky end because it is easy to rejoin the complementary sticky ends. Not all the restriction enzymes make sticky ends, but some cut the 2 strands directly across from one another, producing a blunt end. So if we look at these would be, so EcoR1 would produce a sticky end, and then this ALU1 would produce what's called a blunt end. So I want you to identify what those are. And then exercise 2, it talks about a different part of this packet. So it says, use the restriction map as a circular map. And so then it asks you about if you digest or if you cut in certain spots, how big would the products be? So what would the products of digestion with the 2 enzymes, EcoR1 and EG1? So what I want you to do on that is go to this circle. And so this has base pair length. So this whole circle is 5,541 base pairs long. And so then that asks you if we cut it here and we cut it here, what would our products be? So what I want you to write on the back of this sheet is this would be 1 to 942. So the one chunk would be this big, and then the other chunk would be 5,541 minus 942. So I just want you to give me lengths. So you'd maybe make a picture like this, and then you'd make a picture like this, and you'd say this is 942. And then I think this one's like 4,400 something 95 or something. That's just a guess, but so that's what I want you to do. And so then there's a few other ones, and so then you're going to cut it with Hind 3 and APA 1, and then you'll cut it with 3 different ones, and then it talks about additional products. So the first part is drawing on this, telling me what cut it. So I'd put Eco R1 on this one, and I would say sticky. So do the same thing for all 4 of the strands, and then answer the questions that go along with those. And then on the back, you're just using this little circle and then cutting in the pieces to tell me how long the strands would be. So pretty simple.